Hi everyone, Tony here for Immortality. Every now and then we like to read these papers in full because it's the only way to really understand things. Organoid intelligence, OI, the new frontier in biocomputing and intelligence in a dish. A while back we determined that humans are just not intelligent enough to do what needs to be done in a hypothetical, hyper-futuristic age. And what was required was ASI, artificial superintelligence. While there are certain issues with artificial intelligence, I done artificial intelligence in college. I understand the field and I understand its limitations. I've switched over to biology on the basis of aging and solving aging, enduring aging. And in biology, we have organoid intelligence. Now, organoid intelligence is using biological material, neurons, to build an artificial brain. We're probably at the stage where organoid intelligence is, several thousand neurons, 20,000 neurons, which are brain cells, interacting, forming those networks, exchanging that electricity and those chemicals. We're thinking about a organoid intelligence that encompasses many times the number of all the brains on Earth in a homogeneous, sentient brain that can be communicated with in some way and can be conversed with and can be made to work on building artificial intelligence or artificial superintelligence, with the artificial superintelligence improving the organoid intelligence in a virtuous circle. Because of the hit and miss in sweet spot systems that are impressive because they are hitting a sweet spot, but there is no linear capacity to them. For instance, we would prefer something more like a Moore's law for intelligence rather than a sweet spot limited system, so that each iteration, there's an exponential increase in intelligence, which may not be possible in artificial intelligence. But perhaps if we can have an organoid intelligence that builds an artificial intelligence and vice versa, we might have a kind of projection towards intelligence building intelligence, or intelligence building more intelligence in a kind of Moore's law. New article, Organoid Intelligence, OI, the new frontier in biocomputing and intelligence in a dish, 28th of February 2023, so it's just come out. These are the people who have authored this paper. Let's start. Recent advances in human stem cell-derived brain organoids promise to replicate critical and molecular and cellular aspects of learning and memory, and possibly aspects of cognition in vitro, coining the term organoid intelligence, OI. To encompass these developments, we present a collaborative program to implement a vision of a multidisciplinary field of AI. This aims to establish OI as a form of genuine biological computing that harnesses brain organoids using scientific and bioengineering advances in an ethically responsible manner. Standardized 3D myelinated brain organoids can now be produced with high cell density, enriched levels of glial cells and gene expression critical for learning, integrated microfluidic perfusion systems, can support scalable and durable culturing and spatiotemporal chemical signaling. Novel 3D microelectrode arrays permit high-resolution spatiotemporal electrophysiological signaling and recording to explore the capacity of brain organoids to recapitulate the molecular mechanisms of learning and memory formation and ultimately their computational potential. Now, I say again, this is not to implant brains in people. These are not brain interfaces. And at this stage, the organoids are small, and so their use case is specialized. But ultimately, it's about building a big biological brain. And obviously that takes a whole life support system that goes along with it. With AI, it's chips and silicon and computational power, computational hardware. With a biological computer, it's fluids, chemicals, and a life support system. But if we do it right, that life support system has medical applications. This whole project has medical applications. Technologies that could enable novel biocomputing models via stimulus response training and organoid-computer interfaces are in development. We envisage complex networked interfaces whereby brain organoids are connected with real-world sensors and output devices and ultimately with each other and with sensory organoids, retinal organoids, and are trained using biofeedback, big data warehousing, and machine learning methods. In parallel, we emphasize an embedded ethics approach to analyze the ethical aspects raised by OI research in an iterative, collaborative manner involving all relevant stakeholders. The many possible applications of this research urge the strategic development of OI as a scientific discipline. We anticipate OI-based biocomputing system to allow faster decision-making, continuous learning during tasks, and greater energy and data efficiency. Furthermore, the development of intelligence in a dish could help elucidate the pathophysiology of devastating developmental and degenerative diseases such as dementia, potentially aiding the identification of novel therapeutic approaches to address major global unmet needs. The organoid intelligence, biocomputing, microfluidic system, electrophysiology, cognition, artificial intelligence. Our interest in OI is to build a level of intelligence that could cure aging. But that level of intelligence would revolutionize everything, because society and the society that you live in is an expression of IQ, and it's limited by the expression of IQ, and therefore a superintelligence would issue in a more intelligent age if it were possible. Key points: biological computing or biocomputing could be faster, more efficient, more powerful than silicon-based computing in AI and only require a fraction of energy, knowing that your brain has the capacity to be as smart as a computer, yet it takes much less space than artificial intelligence systems, which are big data centers. So, it's very efficient in the way that in its package. Organoid intelligence, OI, described an emerging multidisciplinary field working to develop biological computing using 3D cultures of human brain cells, brain organoids, and brain-machine interface technologies. Like I said, I'm not really into the brain-machine interface technologies. I'm into building a big brain, maybe a brain the size of a data center that is homogeneous and sentient. OI requires scaling up brain organoids into complex, durable 3D structures enriched with cells and genes associated with learning, and connecting these next-generation input and output devices and AI and machine learning systems. OI requires new models, algorithms, and interface technologies to communicate with brain organoids, understand how they learn and compute and process and store the massive amounts of data they will generate. OI research could also improve our understanding of brain development, learning, and memory, potentially helping to find treatments for neurological disorders such as dementia. Ensuring OI develops in an ethically and socially responsive manner requires an embedded ethics approach where interdisciplinary and representative terms of ethicists, researchers, and members of the public identify, discuss, and analyze ethical issues and feed these back to inform the future research and work. Building a sentient artificial brain is not inherently unethical. It all depends about if its needs are met and if you're putting it through pain. Introduction. Human brains are slower than machines at processing simple information, such as arithmetic, but they far surpass machines in processing complex information as brains deal better with few and or uncertain data. Brains can perform both sequential and parallel processing, whereas computers can do only the former, and they outperform computers in decision-making on large, highly heterogeneous and incomplete data sets and other challenging forms of processing. The processing power of the brain is illustrated by the observation that in 2013, the world's fourth largest computer took 40 minutes to model 1 second of 1% of the human's brain activity. Moreover, each brain has a storage capacity estimated at 2,500 terabytes, based on its 86 100 billion neurons having more than 10 to the 15th connections. In this article, we describe the emerging field that we term organoid intelligence, OI, which aims to leverage the extraordinary biological processing power of the brain. Specifically, both biological learning and machine learning AI by an intelligent agent build internal representations of the world to improve their performance in conducting tasks. However, fundamental differences between biological and machine learning in the mechanism of implementation and their goals result in two drastically different efficiencies. First, biological learning uses far less power to solve computational problems. For example, a larval zebrafish navigates the world to successfully hunt prey and avoid predators, using only 0.1 microwatts, while a human adult consumes 100 watts, of which brain consumption contributes 20%. In contrast, clusters used to master state-of-the-art machine learning models typically operate around 10 to the 6 power watts. Since June 2022, the USA's Frontier has been the world's most powerful supercomputer, reaching 1,102 petaflops, 1.102 exaflops, on the line-back benchmarks. The power consumption of the new supercomputer is 21 megawatts, while the human brain operates at an estimated same 1 exaflop and consumes only 20 watts. Thus, humans operate at a 10 to the 6-fold better power efficiency relative to modern machines, albeit while performing quite different tasks. Second, biological learning uses fewer observations to learn how to solve problems. For example, humans learn a simple same versus different task using around 10 training samples. Simple organisms, such as honeybees, also need remarkably few samples. In contrast, in 2011, machines could not learn these distinctions, even with 10 to the 6 samples, and in 2018, 10 to the 7th samples remained insufficient. Thus, in this sense at least, humans operate at a greater than 10 to the 6 times better data efficiency than modern machines. The AlphaGo system, which beat the world champion at the complex game Go, offers a concrete illustration. AlphaGo was trained on data from 160,000 games. A human playing for 5 hours a day would have to play continuously for more than 175 years to experience the same number of training games, indicating the far higher efficiency of the brain in this complex learning activity. So this is the supercomputers versus the human brain. So it just goes to show you the vast differences in human ability versus natural ability. It's vast. See, if for a supercomputer to reach human brain efficiency, it takes a lot. There was a Hewlett-Packard Enterprise Frontier, or OLCF5, is the world's first exascale supercomputer hosted at the Oak Ridge Leadership Computing Facility in Tennessee. It is compared here with the human brain. The implications is that AI and machine learning approaches have limited usefulness for tasks requiring real-time learning and dynamic actions in a changing environment. The power and efficiency advantages of biological computing over machine learning are multiplicative. If it takes the same amount of time per sample in a human or machine, then the total energy spent to learn a new task requires 10 to the 10 times more energy for the machine. AlphaGo was trained for 4 weeks using 50 graphics processing units, GPUs, requiring approximately 4 times 10 to the 10 joules of energy, about the same amount of energy required to sustain the metabolism of an active adult human for a decade. This high energy consumption prevents AI from achieving many aspirational goals, for example, matching or exceeding human capabilities for complex tasks such as driving. Even large multinational corporations are beginning to reach the limits of machine learning owing to its inefficiencies, and the associated exponential increase in energy consumption is unsustainable, especially if companies are to adhere to their commitments to become carbon negative by 2030. At a national level, already in 2016, it took the equivalent of 34 coal power plants, each generating 500 megawatts, to meet the power demands of the US-based data centers. Being much more energy efficient than current computers, human brains could theoretically meet the US data storage capacity using only 1,600 kilowatts of energy. Notably, the power demands of any current or future implementation of OI is very different from the energy consumption of the human body, especially considering the energy footprint of modern cell culture relative to small organoids today. These comparisons of brains and computers serve only as illustrations of the high efficiency of the human brain. I mean, who cares about that at this stage? You still haven't got proof of concept that you can even contest AI. Together, these observations have created high expectations for biological brain-directed computing as an alternative to silicon-based computing, with the potential for unprecedented advances in computing speed, processing power, data efficiencies, and storage capacities, all with lower energy needs. However, realizing the potential of biocomputing has proved challenging, and most research remains in its infancy. To date, the term biological computing has been used mainly to describe the use of DNA to store digital data. An exception to this is the recent work by Kagan et al., which uses the term synthetic biological intelligence, SBI, to describe the use of synthetic biology to generate intelligent systems through brain-directed computing, albeit using only simple 2D monolayer cell cultures which poorly replicate the complexity of in vivo brain as a proof of concept. I mean, you don't want to just create OI or AI, right? You want OI to be in its native form, that is, It's building those connections in a fluid under a kind of artificial environment which mimics the natural environment. And it's just doing what it's doing, and you're just building that ability for it to become intelligent in its natural state, rather than trying to glue it to a chip. And then you want it to be improving things. You want it to be improving silicon-based AI. You want it to be able to make silicon-based AI more efficient. All right, you don't want to replace it. You want to revolutionize industries with that intelligence. We have coined the term organoid intelligence to describe an emerging field aiming to expand the definition of biocomputing towards brain-directed AI computing to leverage the self-assembled machinery of 3D human brain cell cultures, brain organoids, to memorize and compute inputs. Brain organoids recapitulate organ histoarchitecture and functionality far more closely than the traditional 2D cultures. They can contain myelinated axons and not only show spontaneous electrophysiological activity, but also demonstrate complex oscillatory behavior and exhibit high cell density and layering patterns, all of which make brain organoids superior to traditional monolayer cultures. The question is, can we learn from and harness the computer capacity of these organoids? Achieving this will require major advances in the interfacing of brain cell cultures and computers. We envision using biofeedback to systematically train organoids with increasingly complex sensory input and output opportunities, interfacing the brain organoids with computer sensors and machine interfaces to facilitate supervised and unsupervised learning. We use the term OI for this approach to stress its complementarity to AI, where computers aim to perform tasks done by brains, often by modeling our understanding of learning. However, while AI aims to make computers more brain-like, OI research will explore how a 3D brain cell culture can be made more computer-like. The many possible applications of this work include a new generation of biological and hybrid biological electronic computing technologies, together with advances in our understanding of the physiology of cognition, learning and memory, and the pathophysiological effects of developmental and degenerative diseases, intoxication and infection, which in turn could stimulate drug development and other interventions. OI also has the potential to unlock new neuromimetic AI algorithms with the potential to overcome current AI limitations and aid the development of new brain-computer interface technology. The concept of brain-machine interfacing emerged around 5 decades ago, before the advent of more complex human neuronal cultures and brain organoids. Pioneering work on learning and memory was carried out using primitive animals such as the lamprey, showing long-term potentiation. This led to the brain-machine interaction studies establishing bidirectional communications between the nervous system and external devices. Others used brain slices of different species to study the basic phenomena of learning on a cellular level. Neuron cultures were later shown to perform simple robotic tasks or demonstrate increased plasticity within a delayed closed-loop environment. The combination of brain cell cultures and computers has also been attempted. 2D cultured rat neurons displayed evidence of self-organized activity in a computational task, blind source separation, when applied with electrical information. A different study showed that these cultures learn to respond in a form of distinct electrophysiological patterns to a low-frequency focal stimuli. To the best of our knowledge, however, no relevant approach using brain organoids as learning system has been reported. Previous research has demonstrated spontaneous electrophysical signals and synchronous neural network activity of dissociated organoids, sliced organoids, or developing full organoids, and advanced manipulation of neural circuits within assembloids, organoids merging two distinct brain regions, was recently published. The only study resembling our vision is the recent work by Kagan, which embedded monolayers of cortical neurons in a real-time closed-loop environment via electrophysiological stimulation and recording. These cultures self-organize to rapidly alter their activity to display goal-directed behavior in a simulated game environment. Similarly, the European Union-funded NEU chip project aims to demonstrate the growth of layered networks of brain stem cells on microchips. In addition, the Human Brain Project models similar but virtual input brain output machine models. Obviously, terms such as cognition, intelligence, sentience, and consciousness describing human capabilities cannot be directly translated to simple cell culture models. They are used here to describe the realization of basic functions underlying these high-order functionalities. The workshop to define adequate terminology for the field is in preparation. We see the glossary included, which attempts to provide definitions in the context of this manuscript. In this article, we present in architecture and blueprint for an OI development and implementation program designed to determine the biofeedback characteristics of existing human brain organoids gauged in microelectrode shells, potentially using AI to analyze recorded response pattern to electrical and chemical neurotransmitters and their corresponding receptor agonist-antagonist stimuli. Empirically test, refine, and when needed, develop neurocomputational theories that elucidate the basis of in vivo biological intelligence and allow us to interact with and harness an OI system. Further scale up the brain organoid model to increase the quantity of biological matter, the complexity of the brain organoids, the number of electrodes, algorithms for real-time interactions with brain organoids, and the connected input sources and output devices, and to develop big data warehousing and machine learning methods to accommodate the resulting brain-directed computing capacity. Explore how this program could improve our understanding of pathophysiology and neurodevelopmental and neurodegenerative disorders towards innovative approaches to treatment or prevention. Establish a community of a large-scale project to realize OI computing, taking full account of its ethical implications and developing a common ontology. To the latter point, a community forming workshop was held in February 2022, which gave rise to the Baltimore Declaration towards OI. It provides a statement of vision for an OI community that has led to the development of the program outlined here. Prerequisites for biocomputing models. Culture and bioengineering technologies to advance 3D brain organoids. The advent of two biological technologies make our OI approach possible. The groundbreaking work to reprogram human somatic cells back to stem cells, induced pluripotent stem cells, iPSCs, and the more recent development of 3D brain organoids from iPSCs. So this is its architecture. The architecture of an OI system for biocomputing. At its core of the OI is a 3D brain cell culture organoid that performs the computation, which is probably this thing. The learning potential of the organoid is optimized by culture condition enriched by cells and genes critical for learning, including IEGs. The scalability, viability, and durability of the organoid are supported by integrated microfluidic system. Various types of input can be provided to the organoid, including electrical and chemical signals, synthetic signals from machine sensors, and natural signals from connected sensory organoids, retinal. We anticipate high-resolution output measurements both by electrophysiological recordings obtained via specially designed 2D and 3D shell mirror, and potentially from implantable probes, and imaging of organoid structural and functional properties. These outputs can be used directly for computation purposes and as biofeedback to promote organoid learning, AI, and machine learning are used throughout to encode and decode signals and to develop hybrid biocomputing solutions in conjunction with a suitable big data management system. So what we know is how the brain is used determines the capacity of the brain. We know that from people who have brain injuries, they use their brain more, their whole brain for a single task, and they can do extraordinary things, while a healthy brain compartmentalizes processes to specific parts of the brain, and that's the capacity. So we know that the bigger the brain, the more capacity it has, the more intelligent it is. Advances in 3D organoid culture. The past decade has seen a revolution in brain cell cultures, moving from traditional monolayer cultures to more organ-like organized 3D cultures, i.e. brain organoids. These can be generated either from embryonic stem cells or from the less ethically problematic iPSCs, typically derived from skin samples. The Johns Hopkins Center for Alternatives to Animal Testing, among others, has produced such brain organoids with high levels of standardization and scalabilities, having a diameter below 500 nanometer and comprising fewer than 100,000 cells. Each organoid is roughly 1/3 millionth the size of the human brain, theoretically equating to 800 megabytes of memory storage. Other groups have reported brain organoids with average diameters of 3 to 5 millimeters and prolonged culture exceeding 1 year. Well, neurons can live 100 years. Neurons are probably immortal if they live in a ideal culture, in an ideal environment. These organoids show various attributes that should improve their potential for biocomputing. First, cell density in these 3D models is similar to the in vivo cell density and much higher than in monolayer cultures. The ratio of cells to media volume is also much higher compared to monolayers. Second, most of these brain organoids show spontaneous electrophysiological activity and reactivity to electrical stimulation via evoked field potentials, confirming the presence of active synapses. Trujillo et al. Having shown patterning of cortex layers and oscillation waves comparable to electroencephalograms, EEG, from human preterm babies' brains. Third, axons in these organoids show extensive myelination. Permis et al. Were the first to develop a 3D human brain model showing significant myelination of axons. About 40% of axons in the brain organoids were myelinated, which approaches the 50% found in the human brain. Myelination has since been reproduced in other brain organoids. Myelin reduces the capacitance of the axonal membrane and enables salutary conduction from one node of Ranvier to the next. As myelination increases, electrical conductivity approximately 100-fold. This promises to boost biological computing performance, though its functional impact in this model remains to be demonstrated. So you're creating more of a natural brain in its natural, the way that it is, and that includes axons and myelination, which improve its performance. Finally, these organoid cultures can be enriched with various cell types involved in biological learning, namely oligodendrocytes, mitochondria, and astrocytes. Glial cells are integrally important for the pruning of synapses in biological learning, but have not yet been reported at physiologically relevant levels in brain organoid models. Preliminary work in our organoid model has shown the potential for astroglial cell expansion to physiologically relevant levels. Furthermore, recent evidence that oligodendrocytes and astrocytes significantly contribute to learning, plasticity, and memory suggests that these processes should be studied from a neuron to glial perspective rather than the neuron to neuron paradigm generally used, because it's a network, and that network is constantly changing and being remodeled for efficiency, for optimization. So that's the 2D neural cell, the 3D neural cell with the myelination and conductivity and the improvement of performance that comes with possibly providing a artificial yet mimicking environment to the natural brain, naturally as it exists naturally, rather than trying to make it work artificially, as in the 2D neural culture. That would involve some kind of electrical understanding of the brain as an input-output system. And you're not creating segments. You might be creating segments, but what you're trying to achieve is homogeneity across the entire organoid so that it's all working on one task. And the complexity associated with the brain, it means that who knows what it requires to function correctly. Advances in 3D cell culturing provide the foundation for systems to explore organoid intelligence. A. 3D neural cell cultures have important advantages for biological learning compared to the conventional 2D monolayers, namely a far greater density of cells, enhanced synaptogenesis, high levels of myelination, and encirclement by cell types essential for learning. B. Brain organoid differentiation over time from 14 to 15 weeks, showing neurons. Microtubuli associated protein 2, MAP2, pink, oligodendrocytes transcription factor, olig2, red, and astrocytes, glial fibrillary acidic protein in green. Nuclei are stained with host 33342 blue. Images were taken with an LCM 880 confocal microscope with 20x and 63x magnification. Scale bars are 100 micrometer and 20 micrometer respectively. The images show the presence of MAP2 positive neurons as early as 4 weeks, while glial cells emerge at 8 weeks, and there is a continuous increase in the number of astrocytes over time. In addition, optimizing the cell culture conditions to allow the expression of immediate early genes, IEG, is expected to further boost the learning and memory capacities of brain organoids, since these are key to learning processes and are expressed only in neurons involved in memory formation, as detailed below. Scaling up these 3D organoids is a key early aim. We set out to produce brain organoids with about 10 million neural cells. Existing differentiation protocols for scaling up the cultures and starting 3D differentiation directly from iPSC by passing intermediate generation of neuroprogenitor cells are very promising. These developments benefit from general progress in microphysiological systems, which include organoids, and aim to establish organ architecture and functionality, such as OI are proposed here. Notably, some co-authors are involved in spearheading quality assurance guidings for good cell culture practice, expanding earlier guidance to stem cell-based models, MPS, organ-on-chips models, and establishing an annual MPS World Summit Series and international society. Microfluidic perfusion systems. While brain organoids may recapitulate spatiotemporal molecular signatures, gene expression networks, certain histoarchitectures, cortex patterning, and neuron phenotypes within the human brain, they do not reflect its regional organization and the complexity of its neuronal circuitry to levels allowing for higher order brain functions. Part of the human brain's complexity stems from its size and the vasculature that supports its growth. Although brain vasculature models are under development, most brain organoid models are still avascular and rely on passive diffusion to deliver nutrients. The average scope of diffusion is approximately 300 micrometer before starving. Derived necrosis occurs at the core. Thus, the lack of a perfusible vasculature is a major limitation for improving biological complexity and in vivo-like functionality. Microfluidic systems that substitute for vasculature, allowing controlled perfusion of oxygen, nutrients, and growth factors, and the removal of waste products will be critical to the scalable and durable culturing of brain organoids. These will support the homeostasis and viability of the organoids, allowing a more physiological-like differentiation towards a more complex, sophisticated, and in vivo-like model. Flexible self-folding microfluidics can already deliver chemicals with 3D spatial-temporal control, and recent advances in 3D printing with sacrificial materials offer the potential to create perfusible scaffolds for organoids. These microfluidic systems will also support chemical signaling to organoids. The importance of spatial-temporal chemical patterns to encode information is well established in neuroscience and behavioral science. Significant advances in micropatterning and microfluidics over the past 2 decades have already allowed 2D chips to offer significant tunability of the chemical microenvironment around neurons, and tree-like microfluidic gradient generators have been widely used to create chemical patterns. Importantly, 3D spatial-temporal microfluidic interfaces now enable localized dosing and replication of chemical environments with neurotransmitters, neuropeptides, and other neurochemicals. A comprehensive list of agonists and antagonists is available. Higher resolution recording of cortex neuronal networks, 3D microelectrode arrays for brain organoids, robust and reproducible systems to record electrophysiological outputs from brain organoids are critical to developing OI systems and will need to address various challenges in reading and writing to complex neural assemblies. Brain-machine interface technologies have been in progress for at least 2 decades, but remain primitive. Microelectrode arrays from the heart of many such interfaces, since they can be used to both stimulate and record and offer unprecedented parallelism and individual addressability. 3D microfluidic devices to support scalability and long-term homeostasis of brain organoids. Cells within brain organoids require perfusion with oxygen, nutrients, and growth factors, as well as removal of waste products to provide condition approximating physiological homeostasis. Passive diffusion penetrates to a depth of only around 300 micrometers, and so necrosis occurs at the core of larger organoids owing to starvation. This prevents the brain organoids from being scaled up to size and complexity required for OI research and limits their durability. 3D microfluidic systems enable greater scalability and durability by providing controlled perfusion throughout larger organoids. They also enable 3D spatiotemporal dosing of chemicals and signaling purposes. This is very important. This suggests that this tackles the issue of the life support system for the neurons, and it mimics the body's life support systems that support the body. So it's a very important thing. Now, if you want to scale that up to an unlimited amount, you're going to have to get this right. You're going to have to get the system that supplies the nutrients to all the neurons, clears out all of the junk. You've got to get that going really well. However, most are predominantly in a 2D chip-based format, being designed for use with monolayer cell cultures. This represents a likely problem as brain organoids are spherical 3D structures that make limited contact with a 2D MEA chip. Furthermore, most 2D electrode chip interfaces are rigid, and a mismatch in the stiffness of the recording interface and cell system compromises performance. Like, how are you going to get a big organoid? How are you going to keep saying, all right, this is a bigger organoid, this is an even bigger one, year on year or iteration to iteration? Or how are you going to say, oh, well, we'll get more organoids, make it bigger? Therefore, we and others are developing novel 3D MEA interfaces specifically designed for organoids and inspired by the EEG caps used to record brain electrical patterns from the scalp. Organoids are grown inside flexible, ultra-soft-coated, self-folding and buckled shells covered with patterned nanostructures and probes. This model allows the entire surface of the organoid with unprecedented resolution, high signal-to-noise ratio resulting from a greatly enhanced recording surface areas. After the spontaneous signal is stabilized and synchronized, the response to repeated chemical stimuli from neurotransmitter gradients, glutamate, gamma-aminobutyric acid, GABA, dopamine, serotonin, acetylcholine, and main receptor agonists, antagonists, canic acid, canuric acid, gamma-amino beta hydroxybutyrate acid, GABOB, bisuccoline, haloperidol, nicotine, methyl bromide can be recorded to address the modulate the synaptic plasticity. These shell-mirror interfaces can be integrated with the aforementioned 3D microfluidic systems, supporting the scalability and durability of the system and chemical signaling via spatial patterning and gradients. Together they create a robust platform to gain an iterative, in-depth understanding of organoid behavior and responses to a range of highly modifiable environmental and input stimuli, which in turn will allow us to explore the capacity to recapitulate the molecular mechanisms of learning and memory formation and ultimately their computational potential. High-resolution implantable electrophysiology devices. We consider that the shell mirrors described above strike an appropriate balance by providing comprehensive, high-resolution electrophysiological recordings with minimal disruption to the organoid. However, Future systems might permit organoids to be grown around implantable electrodes to further enhance signal resolution and to access the inside of the organoid. The efficiency of such systems must be balanced by their invasiveness, since any damage to neuronal networks could alter the behavior of the organoid. Neuropixels are silicon probes developed for extracellular recording in animals, mostly mice and rats. They lend themselves to direct integration with brain organoids, though in principle neuropixels can be integrated into shells. These large 10mm dense 100 sites per millimeter implantable neural devices allow the recording of hundreds of well-resolved single neuron signal traces. They can be combined with light sources, electrical stimulation, photometry, dramatically increasing the input and output opportunities and the mapping of the activity in the learning organoid. These devices also appear uniquely capable of long-lived exposure to neural tissue. Chronic implants in rats and mice frequently last for 150 days or more with little degradation in recorded neural activity, indicating suitable compatibility and stability. Recently, the COSIC Laboratory, University of California, Santa Barbara, California, USA, used such CMOS shank probes in parallel with 2D high-density mirrors to record intrinsic network activity in brain organoids. So this would set up the input-output system. And then you would have to take those signals and turn them into some kind of a language. Mounting and inserting such probes into the organoid is a complex challenge, and work is ongoing to develop a new generation of suitable, more flexible probes. If the organoid is grown in well plates, mounting of the probe would be like skull mounting in a rodent. If the growth medium needs to be exchanged periodically for organoid health without disturbing the probe-organoid interface, then an unconventional means for removing and adding media to the wells would need to be developed. The minimum size of the probe base, 4.2mm by 1mm, allows sufficient headroom for this fluidic machinery. Once a microfluidic growth chamber is adopted, a membrane interface to the probe shank will be needed. Neuropixels are routinely used with a quick seal WPI silicon layer over the brain while recording and sealing chronic implants. Further investigations may be required to perfect this geometry, but interfacing with a low-pressure fluidic chamber does not present any fundamental problem. Finally, a NeuroPixels 2.0 probe has 4 shanks, each 10mm long, with a cross-section of 70 micrometer and 24 micrometer. For a 1mm diameter organoid, approximately 500 electrodes would contact the cells, giving the capacity to record from 384 switch selectable sites at a time. The probe electrodes would displace 1.5% approximately of the current organoid volume, likely an acceptable perturbation. Yields of 200 to 600 units are typical in recording from rodents limited by activity. The probe would be within detection distance of approximately 14% of the cells in a 1mm diameter organoid. Among the challenges facing brain-machine interface technology is the scale of connectivity. Cortical neurons each have in the order of 10 to the 4 input synapses and connect to in the order of 10 to the 3 cells, some across many millimeters. Even in small brains such as those of mice, it is not yet clear if organoids have similar or reduced synaptic counts. Current brain-machine interfaces have many unresolved cells per unit reading for each electrode, or within 20 to 80 micrometer, and a largely unknown number of cells for output, writing or stimulation. except for special cases in the visual cortex. The cellular understanding of writing remains difficult, while reading has enabled the control of prosthetic robots. How might traction be achieved to begin harnessing organoid computing power? We propose the two paths be explored. All optical path for organoids would allow cell-by-cell excitation and whole organoid reading, again cell-resolved. While optical imaging is not likely a terminal technology for harnessing OI, it allows exploration of the system behavior and the kinds of computation that could be initially performed. There are rapidly developing technologies for large volume imaging that make this attractive. Techniques such as bezel holography can image volumes with hundreds of micrometer diameter at kilohertz rates with an accurate cellular resolution if the activity is relatively sparse. It's standard EEG, has 25 electrodes, high density there, 250 electrodes, and it reads the output. And that's the microelectrodes around the entire organoid to image the organoid. Currently attainable densities, that's the currently attainable densities. There's some channels, different channels. What did they say? It had 4 channels or something, 3 channels. Interfacing organoids with 3D microelectrode arrays to allow electrophysiological output reading. Organoid mirror interfaces were inspired by the electroencephalograph used to take electrophysiological recordings from the human brain. Organoids are grown inside flexible, ultra soft-coated, self-folding and bucketed cells covered while patterning multi-electrode nanostructures and probes. These interfaces allow ultra-high resolution 3D spatiotemporal simulation and recording of electrophysiological patterns across the entire organoid interface. Bright field image of brain, let's see. Bright field image of brain organoid captured inside the cell. Confocal image showing the side view, projected confocal stack of a brain organoid. Green fluorocalcium dye with a diameter of around 500 nanometers encapsulated in the electrodes of the 3D shell. Blue E and F. 3 channels of electrodes are distributed across the cell with representative raster plots showing the spontaneous electrical activity of the brain organoid. Overlaid spike waveform for the channels 1, 2, 3. So, how do you communicate with the organoid? This is a system of communication. The organoid has shown that the Marcy at San Diego College there, the organoid controlling the aeroplane, and so it programs a different scenario into the aeroplane, and the organoid will correct the aeroplane regardless of the program. So you need an input-output system. Directly writing with opsins is also well established, and holographic methods are again exploited. but the cell rate is significantly lower than the cell read rate. These methods are immediately available and will help inform the design of electrophysiological systems necessary to progress OI. Challenging. High throughput electrophysiology. Electrophysiology recording capacity is embedded by the dark matter observation resulting from the fact that most neurons are not firing most of the time. As such, an electrophysiology channel sensitive to reading and writing individual local neurons can be inefficient. Even the best performing probes see less than 1% of the neurons within their detection range. It seems unnecessary to achieve a synapse level of interpretability of these systems if the base principles are elucidated, empirically tested, and used to control the system as a whole. Notably, bionic implants for humans have managed to convey significant information with relatively few input electrodes. The bionic eye used 24 electrodes, and current cochlear implants use between 10 and 22. Therefore, different resolutions may be required for input versus output. Although organoid EEG and implantable neuro devices are feasible to investigate the scale of required input-output context, both technologies need to be explored further to assess the recording patterns and learning potential of brain organoids. Memory and learning in organoids, training using biofeedback, big data, and AI machine learning. Understanding the capacity of brain organoids to learn is fundamental to determining whether they can be used computationally by harnessing the advantages of biological learning. At this stage, learning is identified as an increased frequency to show and memorize a response pattern to a stimulatory pattern. We aim to use the iterations of technologies described above to interface organoids and computers to initiate supervised learning simulations, trained stimulus response patterns. To achieve this goal, the brain organoids should be exposed to spatiotemporal patterns of electrical and chemical stimulation with the associated recordings, delineating relationship between inputs and outputs. A feedback loop is required to train a learning system. Changes in the brain organoid architecture and functionality, synaptic connections, and electrophysiology due to such training cycles can then be analyzed. These two factors affect synaptic plasticity, the main mechanism of memory formation and learning. Hence, the recorded responses to electrical and chemical stimuli should demonstrate whether and how learning may occur in the organoids. Ultimately, robust high-resolution encoding and decoding of signals going into and out of human cortical tissue is required. Recent achievements using intracortical microstimulation and decoding of motor and sensory cortexes using MEA in human subjects offer promise, though further advances in scale and resolution will be necessary. AI analysis of responses. Organoid mirrors will generate massive recording data sets that will themselves need to be analyzed by statistical and machine learning techniques. Given the recording density and volume, this will necessitate a novel big data infrastructure and supercomputing capacity tailored to the sophisticated needs of this form of modern biological data. Fundamentally, the two major challenges for AI analysis in this context are A. How to decode the inputs provided to an organoid, e.g. the game Pong, to relate to changes with its architecture and or functionality, and B. How to relate these organoids changes with certain outputs, the improvement in playing Pong. In other words, biological computing includes OI as a mediating mechanistic process between the inputs and outputs. To answer these two challenges, we foresee the use of interdisciplinary tools integrating machine learning, statistics, signal processing, information theory, and optimization. We also believe that the questions raised will motivate new methodological developments in these fields. Specifically, we believe the following three paths must be explored to relate OI inputs to outputs. As you can see, this field is in its infancy, and if they're going to support it, then it may be something that might be easy to get in now, but maybe in 20 years' time it'll become so complex that it'll be very hard to get into. Specifically, we provide the following three paths must be explored to relate to OI inputs and outputs. 1. Machine learning and statistical algorithms are needed to quantify organoid function changes. This involves a sensor integration to accelerate processing based on unsupervised learning and dimension reduction, such as principal component analysis, independent component analysis, and autoencoders, including hierarchical versions. B. Signal detection using sequencing and timescales. State space models 131, which are often used in brain imaging analysis. Pattern recognition to identify the real signal pattern and deconvolute it from the background noises. 2. Algorithms are also needed to quantify organoid architecture changes. Challenges include pinpointing the exact parts of the organoid that respond to the input using mixture models, and then quantifying these changes by monitoring their physical appearance. The application of multi-scale unsupervised learning methods to the recorded responses can identify discrete, statistically distinguishable observer unbiased response phenotypes. 3. Models must then be trained to relate to the quantified organoid changes to the output variables via multivariate causal models. OI will require novel developments integrating AI machine learning and both space and time dependent causal modeling. Clearly, inferring or estimating the connectivity of organoids will be a core endeavor. Connectivity in neural circuits is usually divided into structural, functional, and effective connectivity. The distinction between functional and effective connectivity is particularly present here. Functional connectivity refers to the statistical correlations between neuronal fluctuations in different populations, while effective connectivity refers to the causal and directed connectivity between populations. The corresponding data analysis techniques can be passed into frontal connectivity methods such as coherence analysis and Granger causality. These can be contrasted with inferences about directed effective connectivity, usually using some form of dynamic causal modeling. Applying these statistical and computational methods and modern, big and complex data tools such as those from brain imaging and computational biology will allow us to map input and output from organoids' neurological connections. As an example of a relevant technique and fundamentally different approach to neuron behavior mapping was developed in Drosophila optogenetic activation of neurons and the application of multi-scale unsupervised structural learning methods to the recorder responses to identify discrete, statistical, distinguishable, observer, unbiased response phenotypes. So how are you even going to test this stuff? I mean, you're talking about a researcher, trying to improve the system and having just little sample neurons that are kind of test bases for improving all of this stuff using Drosophila, the worm, the fly. So it's kind of lab work, You're going to have, your organoids, and you're perfecting the organoids, you're testing different ways of the organoids, and you're seeing if you've got scalability and if you have something that's more efficient, better way of doing it. And slowly you just get an OI book on all the best current ways of doing things. And obviously, I think that book will be not an advanced book, but like a really early stage book. This could be a starting point for connectivity and achieving and activity mapping studies for further investigate mechanisms through which neurons mediate diverse behaviours. However, with respect to its application to brain organoid recordings, we are entering terra incognita. Machine learning and other mathematical models are increasingly applied to certain parts of organoid research. However, machine learning in the sense of deep learning and supervised learning deserves further comment. This is because it presupposes that supervised learning in an appropriate theoretical formulation of self-organization in organoids, in the sense that the opportunities afforded by organoid research transcend questions about how to engineer a particular behavior through supervised learning. The opportunities probably require a more genetic theoretical framework within which to formalize self-organization and active exchange between an organoid and its external milieu. Practically, if one wanted to train an organoid to do this or that, it would be impossible to implement the procedures for supervised learning in machine learning, i.e. either backpropagation of errors or local energy-based schemes. Current developments favouring reinforcement learning, where self-organisation is met by feedback on functionality, lend themselves to such problems. Strategically, if correct, this means that the direction of travel of organoid research may be either towards reinforcement machine learning or more aligned with some of the foundational questions posed in neurobiology, or indeed the physics of non-equilibrium self-organization. Big data infrastructures. Providing suitable infrastructure for the storage, duration, and processing of OI big data is a scientific challenge of its own. Analytic datasets need to be stored in efficient shared memory structures. Appropriate technology will be needed where manipulations such as standard matrix or tensor calculations can be obtained without partial or streaming access to the full dataset. Furthermore, a fast, robust, and scalable computational analytic and curation infrastructure for the resulting data will be needed. A likely requirement will be efficient dimension-reducing transforms of the 3D sensor arrays applicable for streaming data by scattering transform, which has been particularly successful in analyzing audio streams. Each deep biological network computes some function, transforming inputs to outputs depending on many variables, chief among them the weights and biases and the activation functions. In both AI and OI, it is reasonably safe to assume that the activation functions are essentially static, i.e. any given node of any given time is activated only upon reaching a certain threshold. However, Sinapayan et al. Suggests that neural networks can autonomously change activities to avoid external stimulation. The key difference between AI and OI networks is that in OI networks, the weights and biases may dynamically change over time, for example, owing to growth and or maintaining homeostatic equilibrium. However, with unclear changes in the network function, indeed, vastly different neural networks can implement approximately the same function. A similar result is well established in biological networks. We therefore hypothesize that although the precise weights and architecture of the OI may be dynamic, the memories may be stable over time. The amount of data and respective duration, compression, and processability will dramatically increase with boosted electrophysical recordings from OI systems. Additionally, challenges come from spatiotemporal recordings and the combination of electrophysiology and high component imaging. In addition, the training and experimental data created will require efficient frameworks for storage and analysis. This could include a combination of in vivo proto-neural networks developed in brain organoids and in silico analog digital hybrid and or neuromorphic computing. Transmedia progressive learning will therefore exhibit the advantages of both biological and machine computing and learning while mitigating the limitations of each. The main aspect of our strategy for storage is to develop a scheme resembling the Large Hadron Collider experiment at CERN, where sophisticated triggers are used to detect events in real time and only events with potential discovery value are kept, greatly decimating the data rate. We envisage a similar event-driven way of analyzing the data. We will send discrete stimuli to the brain organoids, look for coordinated responses in many channels, and store only the discrete events related to these intervals. We will use insights gained through multiple versions of experiment reduction to develop a sensor correlation model, optimal event triggers that optimize a trade-off between data reduction and discovery value. Many large ongoing efforts aim to create data processing tools, storage solutions, and standards to handle the scale of data generated by modern neuroscience experiments, e.g. the US Brain Initiative. Open source community solutions which the OI community could leverage are being developed for terabyte and even petabyte scale data across multiple modalities. As high throughput mirrors represent the most promising initial technology for interfacing with organoids, solutions from the invasive and in vitro electrophysiology communities can likely serve as the basis for the OI community. Standardized community processing pipelines exist for this application, including machine learning tools such as data joint elements. Many individual tools and techniques for electrode arrays have been developed for spike sorting and analysis of local field potentials. Community cloud-based archives are available for publishing such electrophysiological data in an open and accessible manner, such as the Open Neuro Archive and Dandy. These archives are hosting an ever-increasing range of data from varied experimental paradigms. Several standard initiatives could be leveraged to facilitate reuse, reanalysis, and meta-analysis, such as the Brain Imaging Data Standard, BIDS, and the Neurodata Without Borders, NWB, by leveraging the standards, processing pipeline, storage, and dissemination techniques developed by the larger electrophysiology community. The OI community can rapidly establish a robust and reproducible big data infrastructure. Looking to the future, additional imaging modalities may become critical to providing further insights into organoid function and learning. Other processing tools and archives exist for relevant modalities such as fluorescence microscopy and also other forms of microscopy, for example, a CAM and pipeline for calcium imaging data analysis, and the Brain Imaging Library for archiving and storage. In the invasive in vivo neuroimaging community, functional and structural connectivity or connectomics is also being studied with improved resolution and larger volumes. Cloud-based processing pipelines for deriving connectivity are under development, and the Brain Observatory Storage Service and Database BOSDB ecosystem has been developed to host and archive large connectivity data sets at the neuron synapse level. Many emerging graph analysis tools enable improved insights such as statistical characterization of connectomics and identification of repeated motifs in time. Multimodal data sets and infrastructure may play an important role in the development of the OI community. Ultimately, the OI community should seek to build on these tools to establish standardized analysis and storage infrastructures. Now, we believe that OI can beat AI. We believe that AI will struggle into the future, and we believe that AI will not struggle into the future. We believe that AI will fulfill the promise that AI should have. AI data sharing can be a powerful approach to grow the community and maximize the reuse of experimental data. Establishing large-scale standardized set of experimental data may rapidly improve processing tools, provide theoretical insight, and generate hypotheses for future experiments. An interesting model approach is the Human Connectome Project, which uses standardized approaches to high-resolution magnetic resonance imaging to provide a gold standard data set for an emerging field of human connectomics. A similar data and infrastructure effort for the OI community can provide invaluable insights. To summarize, a big data ecosystem necessary to study OI will require standardization of experimental data and metadata building on existing standards such as BITS and NWB, robust, repeatable, and standardized processing pipelines that scale to large electrophysiological datasets, efficient, accessible, and open data storage, possibly leveraging existing cloud archives such as OpenNeuro and Dandy, the potential development of multimodal OI datasets, the establishment of standard reference datasets for the community. In the foregoing discussion, we have anticipated a need for the collation, storage, and dissemination of big data under the assumption that organoid research will recapitulate developments in neuroscience and in particular imaging neuroscience. However, there is an alternative path which reflects a move from big data to smart data. In other words, we need only informative data that enables people to answer well-posed questions. In effect, this would represent a pushback against big data and a return to carefully designed experiments that elicit the right kind of data to make inferences about the dynamics, plasticity, and fundamental architectures in brain organoids. In short, the experiences of the neuroscience community may usefully inform the kind of resources needed to realize the full potential of organoid research over ensuing years. Advancing biocomputing complexity. Optimized algorithms for organoid in silico interactions. Realizing the potential of OI requires more than interfacing a computer with an organoid. In OI, the organoid can take on the role of an embodied agent that interacts with an environment through an organoid in silico interface. This will require optimized algorithms for organoid in silico interactions as well as research into theoretical frameworks for learning and adaptations in organoids, drawing from theoretical neuroscience literature. The viability of OI is dependent on optimized algorithms for organoid in silico interactions, in addition to the fast data storage and retrieval described above. There are two broad environments in which OI may operate: open loop and closed loop. Open loop involves feeding information into cells and measuring the response. Recent work leveraging this approach has found that neural systems can alter their activity to perform various tasks, including context-dependent encoding and blind source separation. and to display features such as adaptation towards scale-free dynamics or long-term activity-dependent plasticity. Closed loop extends the open loop environment to include feedback to the neural system about the result of the system activity. Examples of this are limited owing to technical difficulties, but high latency, low temporal resolution analysis suggests that under these conditions, cultures can demonstrate changes in neural dynamics and arbitrary learning. Moreover, real-time implementations have recently resulted in goal-directed learning displayed through a change in neural culture activity when applied in line with neural computational theories. Exploring, applying, and refining empirically supported theories of what fundamentally drives learning intelligence is a critical factor. So far, numerous theories have been proposed to explain how, at a fundamental level, neural systems process and respond to information. The first branch of theories focus on how neural systems are organized both structurally and functionally. Key notions include neural criticality, neural Darwinism, cell assembly, and Herbian plasticity, rule-based learning, and core ideas behind population coding approaches. Generally, these theories aim to explain how the incredibly complex organization within the brain results in its ultimate functioning. Thus, they offer generally compatible frameworks for analyzing and interacting with brain organoids, providing an opportunity for optimized input and decoding of output. Well, at some stage, you're going to have to impress everybody with something, right? Just like ChatGPT was impressive. There has to be some kind of moment of mind-blowing, blowing everybody's mind with some capacity, even though that capacity might not be the ultimate form. A second optimization category of theories generally focuses on how a system or agent works to maintain homeostasis in a dynamic environment. Broadly, this can be achieved either through maximizing a utility or reward or by minimizing surprise or uncertainty. Key theories include the Bayesian brain hypothesis, efficient coding hypothesis, value-dependent learning, optimal control theory, and learning by stimulation avoidance. Attempts have been made to unify these theories recently by exploring the underlying capabilities, most notably through the proposal of the free energy principle, where the system or agent may engage in active inference to construct a generative model of the external environment and act in a manner to minimize the difference between the internal model and the perceived external world. At present, it is difficult to empirically test many of these theories in a controlled manner because in vivo organisms possess compensatory mechanisms that hamper the interpretation of results. OI offers a pathway for highly controlled experiments to empirically test these theories. In addition to frameworks for learning in neural systems, the OI community will require methods to assess embodied intelligence, i.e. computational approaches to understand intelligent behavior in organoids for both open-loop and closed-loop environments. Previously, in vitro experiments have demonstrated the ability of cell cultures to control both physical robotic systems and simulated video games. Experience within the AI community suggests that the OI community will likely benefit from standardized testing environments and conditions, accounting for variability and constraints in the input-output interfacing in terms of channel count and bandwidth. The AI reinforcement learning community has produced a huge range of games, simulation environments, and physical systems that could be adapted to OI. Evaluation. Of particular interest to the OI community may be the field of continual or lifelong learning, where embodied agents are assessed in learning that occurs over a sequence of experiences, often referred to as a lifetime. Such testing environments may serve the AI community by providing important benchmarks for understanding functional activity and learning in organoids. Incorporating complex biological inputs in OI. Previous sections describe how we intend to interface organoids and computers, combining organoids with various types of more complex inputs and output stimulation and recording interfaces will allow us to understand the potential for real-time controls. The consequences of such interconnections can be explored starting with two brain organoids, one with complex input and one with complex output connections. The sensory organs, such as a retinal organoid, could then be connected with a brain organoid. Eventually, networks of organoids will be interconnected to implement more complex OI. The organoid will be interfaced with electrical and fluidic sensing and simple outputs controlling machines through biofeedback on the cellular level, giving the brain organoid control by feeding back the results of its induced actions. By connecting retinal and brain organoids, we can determine whether signals can transfer between these different neuronal organoids and how this exogenous biological signaling will be interpreted by the brain organoid, establishing the initial baseline of organoid communication. Retinal organoids containing laminated retinas with outgrowth of outer segment-like structures and synaptic ribbons have been developed. Synapse formation between two organoids is complex and was demonstrated recently in assembloids. Retinal and brain organoids can be connected either through electrodes or directly. Since methods to generate mature endogenous light-sensing retina are preliminary and very inefficient, engineered photosensitive ion channels, which are expressed under the control of photoreceptor-specific promoter, can substitute as a proxy for light reactive photoreceptors. Optimization of retinal organoid culture conditions to promote more robust signaling have recently been published. Understanding and perhaps even influencing the connectivity of retinal and brain neurons would be extremely exciting. Although we and others are actively investigating ways to establish and modulate these connections, we are still quite far away from a system that can demonstrate robustly modifying neuronal conditions in a directed manner. Ultimately, we aim to build on this simplified approximation or representation of visual input towards a system that more fully approximates vision. We're going to give the organoid eyes, maybe try and give it a mouth, teach it language, give it eyes, which is massive brain, like the size of a building, and just eyes and a mouth. Leveraging advances in the molecular basis of biological learning. Advances in the molecular biology of synaptic plasticity will be critical for optimizing the capacity of organoid systems for learning and OI. Growth conditions can now be optimized to allow organoid neurons to optimally express genes essential for learning in the human brain. First, they need to express genes coding for the neurotransmitter receptor that mediates synaptic transmission. We have already characterized the expression dynamics of different subunits of the N-methyl-D-aspartate NMDA glutamate receptor during differentiation, unpublished observation. Long-term organoid maturation and age based on gene expression and switch of the main receptor subunits were recently extensively characterized. The machinery of the organoid biochemistry, especially the neurotransmitter, will be an important part of understanding the signaling cascades and synaptic plasticity necessary for learning. To this end, a thorough characterization of protein expression is warranted. Secondly, it will be important for organoids to express IEGs. IEGs mediate synaptic processes essential for memory consolidation, and are rapidly transcribed in adult neurons as they process information. Moreover, evidence indicates that IEGs are required for circuits capable of gamma frequency rhythmicity, which is associated with attention and information processing. Using RNA sequencing to compare organoids at different stages of maturation and grown under different cultural conditions with fetal, adolescent, and adult brains will allow the identification of growth conditions that optimize expressions of these essential genes. It is critical to ensure IEG expression that is robust and dynamically responsive to actively evoked by a pharmacological block of GABA inhibition, e.g. using biocomputing. Dynamic activity-dependent IEG expression would assure the presence of molecular substrates necessary for organoid to establish circuits with balanced excitatory, and inhibitory neurons and synapses. The cell culture conditions should be optimized so that 10 to 30% of neurons express IEGs in response to informational inputs. This level of sparsity is typical of brain regions involved in learning and memory, including the hippocampus and neocortex, and may ensure that multiple assemblies can be created to uniquely encode different streams of data. This contrasts with the global activation of IEGs that may occur with non-informational activities such as seizure. IEG expression and/or associated reporters can also provide a means to assess the formation of assemblies of neurons that are stably linked to specific inputs in the brain. Such assemblies and thought to represent memory engrams. EEG data can then be correlated with dynamic activity reporters, CA2 plus or voltage, and IEG expression data. Ultimately, neuronal activity can be stimulated and recorded optogenetically, as recently described. To our knowledge, IEG expression has not previously been utilized as an endpoint for optimizing organoid growth conditions. Successful use of this parameter would establish a biological basis for OI and address concerns regarding the uncertainty of the developmental state of organoids and their potential utility as information processing memory units. OI led advances in medical research and innovation. In addition to pioneering the use of human brain organoids for computing and learning, OI research will also allow the exploration of inter-individual neurodevelopmental and neurodegenerative differences between stem cell donors. Alzheimer's disease and other dementias could represent a particularly important priority for research. Globally, more than 55 million people are living with dementia, and this number is projected to exceed 150 million by 2050. Dementia is among the top 10 leading causes of death and globally costs at least $1 trillion annually. What if the organoid starts getting Lewy bodies in them and starts to infect the whole thing? What if the organoid gets Alzheimer's disease? You can't have that. Clinical trials of novel Alzheimer's disease therapies have shown very poor success rates, in part owing to premature translation of successful results in animal models that mirror only limited results. Aspects of the pathology in humans. The adaptation of OI research models to neurodegenerative diseases would offer the first human-based preclinical model to help us understand and develop effective treatments for these devastating diseases. In addition to neurodegenerative diseases, neurodevelopmental disorders also lend themselves to AI, given that the different stages of brain development are reflected in the bioengineering of these models from stem cells. Conditions such as autism are major concerns owing to this enormous increase in Prevalence in the US autism spectrum disorder was diagnosed in the 1970s and 1 in 10,000 children. But according to the Centers for Disease Control, in 2021, it was 1 in 44. While the disorder has heritable aspects, environmental influences are an increasing focus. The brain organoid model has shown promise for both developmental neurotoxicity and gene times environment studies of autism. A varying combination of cognitive impairments characterizes this complex spectrum of diseases. Similarly, leukodystrophies are a diverse group of rare genetic disorders affecting the white matter and linked to cognitive impairment. Using OI to explore the genetic basis of autism or leukodystrophies appears to represent an important path to understanding these disorders and to allowing screening of potential drugs that might boost underdeveloped cognitive functions. Schizophrenia affects around 1% of the population worldwide and is one of the top 10 illnesses contributing to the global burden of disease. Schizophrenia has a prominent genetic basis, and it has been suggested that it may be neurodevelopmental in origin. Prenatal complications are an important contributor to the condition. While cognitive dysfunction is a hallmark with a strong similarity to autism, human iPSC lines, e.g. with genetic backgrounds associated with disorders, are available and continuously growing. SFARI base for autism. Organoids developed from iPSCs from individuals with various conditions could be compared against control samples to help identify a difference that may elucidate the pathogenesis, risk factors, and treatments. The application of an OI approach using these cell lines would thus be very promising to aid further understanding and characterization of the etiology of the neurodegeneration, neurodevelopmental, and psychiatric disorders. This enables a multitude of applications for de-risking pediatric drugs for adverse effects on cognitive development, the identification of toxicants and illicit drugs with long-term effects on cognitive capabilities, and the optimization of lead drug candidates acting on respective pharmacological targets. So they want to use these, or you can just develop these organoids and you can test stuff on them. Like they represent a brain model that you can experiment on. Like, how does lead affect them? How does these toxicities affect it? But that's not what we're after. It's not OI. OI is a competitor to AI. We're trying to beat AI. Trying to create an organoid or an artificial or a biological brain in a vat that is more capable than AI. The study of memory, learning, and cognition and the impact of neurodegeneration on these functions will require physiologically relevant neuron to glial ratios and higher levels of biological complexity and interregional communication. Besides assembloids, aspects of ventral and dorsal region in the absence of external morphogens or growth factors have been recapitulated, highlighting an innate ability of self-organization in both cases. Recently, Cedarquist et al. Demonstrated that a chimeric assembloid of an early organoid and a cluster of sonic hedgehog SHH secreting cells resulted in dorsal, ventral, and anterior posterior positional access. However, organoids do not have predictable anatomy nor defined topography, and generally do not reflect the characteristic developmental asymmetry. Nonetheless, brain organoids do show a remarkable self-organizational capacity. This might be further enhanced by functional demands, which may be harnessed when scaling up the size and inducing regional polarization, thereby increasing the biological complexity of neuronal networks and interregional connections to better reflect brain architecture and function, ethics of biological computing with organoids. Creating a human brain model with inputs and output as well as learning capability raises complex ethical questions. At 12 weeks of fetal development, the human brain has a weight of approximately 3 grams, a volume of approximately 3.5 milliliters or 3.10 to the 9 cells in the neocortical part of the fetal telencephalon. An adult mouse brain weighs approximately 0.4 grams. In comparison, current brain organoids have a diameter below 500 nanometers in culture with less than 100,000 cells. The maturation of the brain organoid is accelerated by growth factors so that at 10 weeks of culture, organoids show some features such as myelination that start in the fetus about 20 weeks of gestation. Furthermore, the stimulation with information input might lead to very different organoid development, and much longer culture periods would be envisaged for training organoids, together possibly augmenting cognitive capabilities. The ethical concerns raised by brain organoid research have mainly focused on questions about creating entities that could potentially exhibit consciousness. Could organoids experience pain, and if so, would they suffer even in rudimentary ways? These concerns will mount during the development of OI as the organoids become structurally more complex, receive inputs, generate inputs, outputs, and at least theoretically process information about their environment and build a primitive memory. This will require deeper analysis and research regarding the morally salient neurobiological features that contribute to human capacities, including consciousness, and the implications for OI research and the implementation when some of all of these are met. Articulating the physiological conditions that are necessary and sufficient for consciousness is one of the most difficult puzzles of neuroscience. To assess whether organoids exhibit the criteria for consciousness will require some consensus to be reached about what those criteria are. Work underway to uncover the neural basis of consciousness will inform the evaluation of the ethics issues raised by OI. However, it will also be important to distinguish consciousness from sentience, formerly considered as awareness to stimuli. Response to sensory input. Such use of terminology could be debated and represents a critical challenge for forming OI community. We use the term sentience in the most basic way, similar to the way in which many aspects of cognition have to be understood at very basic cellular mechanisms, not human-level brain functions. Even recent proposals for the use of the perturbational complexity index (PCI) assume the more complex idea of phenomenological consciousness when the behavior could be explained by simpler sentience. Notably, the suggested OI program does not aim to recreate human consciousness, but rather functional aspects related to learning, cognition, and computing. Well, it depends how much money it has in its bank account, really. If OI is billionaire, maybe it'll be happy. Nevertheless, as advances in the structural and functional complexity of AI systems begin to recapitulate aspects of human neurobiological subprocesses such as learning and cognition, researchers will inevitably encounter the Glial dilemma, a situation whereby incremental successes in modeling aspects of the human brain will raise the same kind of ethical concerns that originally motivated their development. Sufficient advances in OI will raise questions about the moral status of these entities and concerns for their welfare. Frameworks have been proposed to address these ethical concerns in research practices, but it remains unknown whether these proposals adequately attend to the moral concerns held by the public. For example, harm reduction policies are often unsuccessful in gaining public support when the underlying attitude is based on a moral conviction with implication for public discourse. I mean, how bad are some human beings being treated, let alone something that's going to live in, it's going to have everything provided for it, and, and all the dopamine at once, whatever. That was a dumb thing to say. You're going to have this thing that's going to have all of its nutrients taken care of. It's going to live in luxury. It's going to be taken care of. All of its needs are going to be met. Versus some people in society who are treated badly, and no one says, well, that's unethical that they're treated badly, but that's just normal. They've accepted that as normal. Comprehensive ethical analysis of OI will require input from diverse publics and relevant stakeholder groups in order to, 1, prevent misunderstandings from creating unintended moral appraisals, and 2, to foster trust, confidence, and inclusion through responsible public engagement. Well, think of it, if all your needs are met, would you care? The answer is no. Notably, moral attitudes towards OI may depend less on epistemological concerns mentioned above, such as the role of specific cognitive capacities in assessment of moral status, and more on ontological arguments on what constitutes a human being. Perceptions of recreating human-like entities in a lab are likely to invoke concerns about infringing on human dignity that could reflect secular and theological beliefs about the essential nature of the human being. Our approach to embedded ethics in OI will seek to identify and attend to these ethical concerns by informing future public engagement and deliberation on OI. Well, I envisage like one OI, one big OI. I don't envisage millions of OIs. I just envisage like this one super project by, or a handful of them by a handful of companies that are just tasked with producing this huge intelligence and then putting it to task to slowly reinvent the world, including curing aging, including doing the physics and the science work. Our approach to embedded ethics in AI will seek to identify and attend to these ethical concerns by informing future public engagement and deliberation on OI. Other issues anticipated to require attention include privacy concerns on the part of iPSC donors and aspects of intellectual property. What does the organoid exhibit about its cell donor? Is there a moral obligation to inform the donor if, for example, something relevant to their health is identified during research? Do donors have the rights that extend beyond the donation? In common with other scientific and bioengineering aspects of our eye, this is truly uncharted territory. The ethical considerations and viewpoints can be expected to evolve with an increased understanding of organoid systems. It is therefore critical to frame the ethical consideration at the onset of this research in a manner that encompasses all anticipated issues and which continually reflects on progress and new lessons. We propose to use an embedded ethics approach whereby an ethics team will identify, discuss, and analyze ethical issues as they arise In the course of this work, embedded ethics is a standard approach in interdisciplinary ethics research whereby expert ethicists join and collaborate integrally with research and development teams to consider and address ethical issues via an iterative and contiguous process as the research evolves. Box 1 offers a preliminary framework of ethical considerations informed by discussion at the first Organoid Intelligence OI workshop to form an OI community workshop, which was 22-24 February 2022. While the embedded ethics approach provides a mechanism for investigating the physiological and scientific conditions relevant to the moral status of brain organoids, it has no inherent mechanism for seeking, identifying, or incorporating public values in the development of OI. So this is Box 1, the preliminary framework of ethical considerations in organoid intelligence research. Organoid intelligence research entails many important ethical aspects that warrant an iterative, collaborative ethical process as the field develops, involving all relevant stakeholders. Here we offer a preliminary framework of issues for consideration: cell donation and the potential issues, bioculturing and the potential issues, organoid-machine interface and the potential issues, learning and computation and the potential issues, and applications and the potential issues. It is important to understand public perceptions of OI, and this cannot be delegated to ethicists alone. It needs to be embedded within the field as a three-way feedback loop involving research, ethicists, and members of the public, including stakeholders who could be especially impacted by advances in AI, neurodiversity advocates. This feedback loop will enable specific applications of OI to be articulated by researchers, analysed by ethicists based on theoretical principles, and evaluated by members of the public with diverse moral perspectives. The views expressed by the public then inform the work of both scientists and ethicists seeking to advance OI in a socially responsive manner. This calls for public dialogue has been echoed by a National Academy of Sciences report on human neural organoids, the recommendations of innovation in neurotechnology by Organization for Economic Cooperation and Development, and various neuroethics committees. Moreover, researchers in science, communication, and deliberative democracy have demonstrated that deliberative techniques are one of the most effective mechanisms for informing the public and mitigating the risk of polarization on contentious issues. Finally, public engagement on OI will not only be necessary to prevent adverse public reactions, but will also maximize the future impact of the field and serve as an exemplar on how to embed society within science. Of relevance in this context, co-author J.K. co-chaired the Neuroethics Subcommittee for the new strategic plan for NIH Brain 2.0. The only project we know of with relevant parallel aspects is the Brainstorm project led by In-Soo Hin at Case Western University. We will be further actively promoting discussions between biologists, ethicists, and philosophers who are interested in brain organoids and navigating brain organoid ethics. In research, as we did before, a recommended way forward would include an agreement on commonly used language, the need for research on the neural basis for consciousness as above, and 3, the development of the best practice guidelines that consider the views of the relevant stakeholders. Conclusion and action plan for OI research. We present a collaborative, iterative, a multidisciplinary program aiming to establish OI as a form of genuine biological computing that harnesses brain organoids using the scientific and bioengineering approaches described here in an ethically responsible manner. Ultimately, we aim towards a revolution in biological computing that could overcome many of the limitations of silicon-based computing and AI and have significant implications worldwide. Specifically, we anticipate AI-based biocomputing systems to allow faster decision-making, including on massive, incomplete, and heterogeneous datasets, continuous learning during tasks, and greater energy and data efficiency. Furthermore, the development of intelligence in a dish offers unparalleled opportunities to elucidate the biological basis of human cognition, learning, and memory, together with various disorders associated with cognitive deficits, potentially aiding the identification of novel therapeutics and approaches to address major global unmet needs. So this is the action plan. Trajectory 1, scale up in vitro brain organoid electrode computer infrastructure interface. Trajectory 2, biocomputing with brain organoids. Trajectory 3, brain organoid machine and organoid interactions. And trajectory 4, ethics of biological computing with organoids. So it's not a roadmap, it's more like 4 parts of the same thing. So I like the fact that they said larger brain organoid cultures with nutrient and oxygen perfusion, allowing long-term culture. So big brain, bigger and bigger and bigger. Smarter and better. I mean, it's gonna be a sight to see. I mean, this thing gets to a certain size, and you see all of the systems. You know, these huge systems around this small thing, this thing that fits into a jar. You're just gonna have these huge systems around it, and just trying to just really be nice to it, and trying to really, work it properly. Okay, so the conflict of interest is TH is a named inventor on a patent by Johns Hopkins University on the production of brain organoids, which is licensed to Axosim New Orleans, LA, United States, and receive royalty shares. TH and Alice consult Axosims. JS is named as inventor on the patent by University of Luxembourg on the production of the midbrain organoid, which is licensed to organotherapeutics. So there is some conflict of interest. Cortical Labs. Do you remember Cortical Labs? It's recently showed some results of their organoid glossary. Organoid intelligence, OI, the new frontier in biocomputing and intelligence in a dish. Will it beat AI? Will they ever get it to a stage where it gives the latest AI a run for its money? And what a sight that will be. If we get a tour of it, we'll see that small organoid, and we'll see these huge systems around it just keeping it alive and processing the data that it's producing. And the idea is that we exceed human intelligence through one of these methods, and we cure aging and disease with it. Tony, over and out.